Hello and welcome everybody to today's webinar of the Permeability series. We are waiting a couple more minutes, like until 5 past, before we start for everyone to arrive. And yeah, looking forward to it. Okay. Aber jetzt trotzdem nicht länger warten. Oder? Also jetzt noch einfach. Great. So now we've reached the 5 past mark, and I think we're good to go and start. So again, hello and welcome everybody to the next part of the Permeability series. Today's topic is about the estimation of oral absorption from self-nanoemulsifying drug delivery systems, SNEDDS in short, using an in vitro lipolysis permeation method. My name is Ali and I will be the moderator for today's webinar. We are very happy and proud to have 2 new presenters talking about their experiments and researches on permeation. First, we'll have Ragna Bertelsen. She's an associate professor at the University of Copenhagen in Denmark at the Department of Pharmacy since October 2020. Ragna has researched, among other things, on drug delivery and development of predictive models for the gastrointestinal tract, especially for poorly water-soluble compounds. She also held lectures and keynotes on different topics regarding drug formulation and bioavailability. Next to Ragna, we'll have Mette Klitgaard, With us, Mette is a PhD student also at the Department of Pharmacy at the University of Copenhagen with a master's degree in pharmaceutical sciences. She has told us to have a fascination for in vitro models estimating oral absorption of drugs since her undergraduate days. Her PhD project focuses on the study of the mucous layer of small intestine and its effect of the absorption of poorly water-soluble drugs. This shortly wraps up Rauna's and Mette's academic career so far, and we could not think of two more fitting presenters for today. Before we start with the webinar you have been all waiting for, a few things regarding the organization. We have opened a Q&A window where you can type in your questions. We will answer them at the end of the presentation, and we'll also provide the slides and a link to the recorded webinar at the end of the presentation. Without further ado, I give the word to Rauna and Mette, and enjoy the presentation, and see you at the Q&A again.
Thank you very much, Julie, and thank you for the very kind introduction and for the invitation to be part of this webinar series. I hope you can all hear me well. The agenda for this presentation is that I'm going to start off introducing you to the use of in vitro models to estimate in vivo performance of orally administered drugs. After giving this very general introduction and why we use it and what we have to take care of, I'm going to also introduce you into lipid-based drug delivery systems, and then I'm going to give you a brief introduction into the typical in vitro models we use when we are estimating the performance of these lipid-based drug delivery systems. Following this more general introduction, Mette is going to take the stage, and she's going to give you an introduction as to the model development work we've done, developing an in vitro model that combines lipolysis and permeation. She's going to guide you through our results and the correlation to in vivo in vitro correlations. So when we're talking about using in vitro models to predict drug absorption, why do we actually use these models? Well, as Max has also told you previously in these lectures, it's really to reduce the cost and time of the preclinical drug development. When we're using in vitro models, we can provide input for our in silico drug profiling, and we can really refine or especially replace and reduce the animal testing. And so that really shortens this cost and time period for the preclinical drug development. Another advantage of using the in vitro models, and what we do a lot also here at the university, is to use the models to increase our mechanistic understanding of what's actually going on when these drugs are orally administered and absorbed to reach the systemic circulation. When we're using in vitro models, what we have to keep in mind... Sorry?
Sorry, everything fine.
Good. Then I'm going to continue. I got confused. But what we really have to keep in mind when we're using these in vitro models to predict drug absorption is that we're using a very simplistic system, or at least commonly something very simple like a dissolution vessel or a simple permeation setup to model something that's really very complicated. Because after we are ingesting our medicines, it's going to reach the gastrointestinal tract where many different processes are occurring, or many different physiological concepts are occurring at the same time. There's going to be an interplay of a lot of different things that's very complicated. And we're trying to mimic that in a very simplistic manner. The way we're using the in vitro models, these simple models, is that we're trying to model the rate limiting step to absorption. So that is like modeling one part of everything that's going on after we ingest our medication, and saying that that's the most crucial part to mimic in order to estimate how fast our drug is going to reach circulation. So with this in mind, it's really important to remember when you are selecting what type of in vitro model you're going to use. And it's also really important to remember what sort of assumptions you already made when you're interpreting the in vitro data that you get out. Yes, so that is really important to keep this in mind. So what do we actually mimic when we are using in vitro models? Well, in general, we can split it up into two different types of processes. We look very much at the drug solubilization or drug release, and then on the drug permeation. In order for a drug to have a systemic effect, it needs to first be in solution and then to cross our intestinal barrier after oral absorption. Therefore, we generally split our in vitro models into solubilization or release models that can look into the disintegration or dissolution process if we're looking at solid drug delivery systems, or looking at the digestion process if we have something digestible like a lipid-based drug delivery system. Aside from splitting it into these general classes of in vitro models, it's also really important to know a lot of information before you select or design your in vitro models. It's important to know something about the drug substance, the physical chemical characteristic. That's going to tell us about to what degree do we actually need to mimic the physiological media and the different barriers that the drugs might interact with. Also, it's very important to know what type of drug delivery system we have, what expected excipient effects might be relevant, and then that's going to help us also determine what type of models do we need to use. And lastly, it's also really important to know something about the physiological characteristics of either the specific target that you are aiming for in the gastrointestinal tract or a specific population that this drug delivery system is targeting. It's very different if you're simulating, for instance, a pediatric population compared to an adult population. So therefore, that it gives you different considerations when we're developing our in vitro model. So this is really just stating a lot of things of that in vitro models are really important and we can use them for a lot, but we also have to really take care on how we design them and how we interpret the results. That being the general in vitro model description, I'm going to continue on giving you a brief introduction to the lipid-based drug delivery systems. These drug delivery systems are systems that has been developed for the poorly water-soluble compounds that are generally very lipophilic. So that is drugs that generally have a higher bioavailability if we co-administrate them with food. In order to do this in a very controlled manner, we can dissolve our drug in a lipid-based formulation. So that's a formulation containing oils, surfactants, and co-solvents. And then we can administrate our drug in solution. Thereby, we're going to bypass the slow dissolution step for these poorly water-soluble compounds and administrate the drug in solution. When it's in this lipophilic solution, what's going to happen is it has to be released from the lipid base. So that means it's going to there's going to be a transfer between the different digestion phases, the colloidal phases that are formed, and then an absorption through the intestinal barrier. What we also have to keep in mind is that the whole point of using these lipid-based drug delivery systems is to give the drug in solution, and therefore it's very critical that the drug doesn't precipitate, or at least it doesn't precipitate crystalline, because then we're sort of back to starting if we'd had a solid-based system. In terms of the different classes of lipid-based drug delivery system, it really depends on what type of excipients are used for these systems. So we can have very simple formulations like an oil solution. So that's just selecting specific oils or lipids and dissolving our drug. Then we already prepare them as emulsions or microemulsions, as liposomes, solid lipid nanospheres, or we can have these self-nano or microemulsifying drug delivery systems. Since this is the type of system that will be presented later, I also just brought the definition of a SNEDDS, and that's defined as an isotropic mixture of natural or synthetic oils, solid or liquid surfactants, or alternatively one or more hydrophilic solvents or co-solvents that have the ability of forming a fine oil and water emulsion upon mild agitation. So that is, we dissolve our drug in this mixture of oil, surfactants, and co-solvents. We administrate that, and when it reaches the gastrointestinal tract and whatever fluid is in our stomach or small intestine, we will have this self-emulsifying system. So we'll have a formation of a very fine emulsion where the drug can be released from. So after we administrate this drug, then we will want the drug to reach the blood circulation. And if we want to in vitro evaluate how this drug is going to work, we are going to first need to have some knowledge about what actually happens after we administrate the drug. And when we administrate the drug orally, it's going to go through our mouth and our esophagus down to our stomach. In our stomach, we're going to have an initial digestion process. Mainly, it's going to be mechanical by grinding because we have contractions, especially in the antrum part of our stomach. So the formulation is mechanically digested, which is generally going to mean that we have a formation of smaller emulsion droplets. And then also an enzymatic digestion will be initiated since we have acid and also gastric lipase present in the stomach. After the formulation transfers into the duodenum and the small intestine, we'll have bile and pancreatic fluid added as well, which is really going to initiate the enzymatic digestion process. And this is, again, many things occurring at once. But when we are digesting our lipid emulsion droplets, we're going to have the formation of different colloidal structures. We're going to have small micelles and mixed micelles where the bile and whatever free fatty acids are released here are going to go together and form these different colloidal structures where our drug can be solubilized inside. So during digestion, some drug is going to be partitioning into the aqueous media. Some drug is going to be in these different colloidal structures. And all the drug, due to the concentration gradient, is going to transfer towards the intestinal area. When the drug is released from this lipid formulation, it generally forms a supersaturated solution, since the concentration in our lipid phase is much higher than the equilibrium solubility in the aqueous phase. From this supersaturated solution, our drug can then precipitate. It can precipitate crystalline, and then it will usually have a slow redissolution process before the drug can be absorbed, or it can precipitate in an amorphous form where we usually have a faster redissolution process before the drug is absorbed. So this is all the release mechanisms occurring. But then after the drug is released and has transferred through this unstirred water layer, the drug has to permeate the enterocytes. And again, the permeation heard before can occur in many different ways. So we can have the passive diffusion either paracellularly or transcellularly, but there can be some active carrier involved. So we can have either the drug transported through the cell layers, or we can also have an efflux again. So there can again be many different things occurring here during the permeation process before the drug actually reaches the blood. So as you can see, a lot of things happen after we ingest a lipid-based drug delivery system, but then how do we mimic that in vitro? The most common thing to do is actually to say, we're only just going to focus on the first part here. Because these drugs are poorly water-soluble, often class 2 compounds that might permeate relatively easy, and therefore the most important thing is to mimic how the drug actually goes into solution. Yes, so if we're mimicking how the drug goes into solution, the general setup that has been used for the past 20, 30 years, I think, is this pH stat controlled lipolysis setup, where we have a thermostated reaction vessel simulating our stomach or small intestine, usually just the small intestine. In this vessel, we're going to have biorelevant media and digestive enzymes, and of course the formulation we have. To the medium, we can add calcium, which is going to precipitate whatever free fatty acids are released during digestion, and this means that we have a continuous digestion process. And that sort of simulates the in vivo process, as these free fatty acids are usually absorbed, allowing the continuous digestion process. We also have a pH probe and a titration of sodium hydroxide, meaning that we can measure the pH continuously, because the digestion usually means that the pH is going to drop because the acids are released, the free fatty acids, but then we can keep the pH constant, similar to what we see in the small intestine, by adding sodium hydroxide. With this pH control, we also have the ability to measure how much digestion is occurring. So we have a tool to sort of interpret the results and link the digestion process to the drug release. And that means this is a good setup for getting a better mechanistic understanding about what's actually going on during this digestion and drug release process. During a setup here, we usually take out samples and centrifuge them, and then we have this phase separation. We will usually have a pellet phase, where of course we have the precipitated free fatty acid or calcium soaps. If there's any drug that's precipitated, that will also be in the pellet phase. Then we're going to have an aqueous phase that contains all the aqueously solubilized drug and all the colloidal structures. And then if there's a lot of undigested lipids present, they're going to form this lipid phase on top. When we're using this in vitro setup to estimate something about the in vivo performance of lipid-based drug delivery systems, we generally assume that whatever drug is in the aqueous phase here is directly available for absorption, and assuming that the permeation across the intestinal barrier just occurs, then we can link whatever is in here to whatever reaches the circulation. Um, with that assumption, we can sometimes have very nice in vitro correlations where we can rank order whatever amount of drug is in this aqueous phase. So here's a study where danosol is in a lipid-based formulation of 4 different lipid-based formulations. And you can see that there's different amounts available in the aqueous phase. So based on this in vitro output, we can rank order saying that clearly that the diamonds here has the highest amount of drug in solution in vitro, which we're going to assume means that we have more drug available for absorption, and therefore we would predict it would have a higher bioavailability compared to the other formulations. And if we look at the plasma profiles obtained here, we can see that the diamonds did in fact have a higher Cmax and a higher AUC, linked to a higher bioavailability compared to the other formulation. So in this case, we can see we had a nice correlation and that we can use the in vitro setup we already I just presented. However, in other cases, and I think by now it's about the equal number of cases, when we do this in vitro lipolysis setup, then we can see that here we have now phenotype-based lipid-based formulations, and this is the amount of drug in the aqueous phase. And in vitro, we here see that the blue formulation clearly outperforms the green and the red, with the black at the very low. In vivo, we see a different trend. Here we see that the green and the red are outperforming the blue and the black. And if we would plot it as in vivo AUC over in vitro AUC, you see there's no correlation here. So in this case, we wouldn't actually be able to use our in vitro predictions to select the best in vitro, the best formulation in vitro, because that doesn't correlate with in vivo. So with these the lack of in vivo in vitro correlation, that has really left us modeling agents with the question like, what do we do? Because if we cannot use the in vitro digestion models to estimate what's going on, then how can we change this model design? And in general, there's been different tendencies during the past up to 5 years or something, most interest in the last couple of years. What's been done the most is people suggest it's because we're lacking this permeation module. There's no absorption occurring. We are not simulating the intestinal barrier, and therefore we might see a wrong picture, especially in the cases like I described, where we have drug supersaturation. So we might have something in a high concentration that has a tendency to precipitate. If it doesn't get absorbed, then we might actually get a really wrong interpretation of what's going on in vitro compared to in vivo. So there's a lot of focus on combining digestion and absorption to estimate the in vivo outcome. And this is also, that's why it's in bold. This is what Mette going to talk to you about in a very short while. But just to say, another trend that's been seen is that taking care of this species differences, because in the setup that I just showed you, then commonly we are simulating the human digestion. So we have media simulating the human intestinal fluid and activity levels, enzymatic activity levels, that simulates human digestion. But then when we are doing our preclinical studies, it's usually done in animals. And in the examples I've showed you, it's been done in rats. And therefore we have a species differences. So another thing that has been done and also done in our research group is actually to change the in vitro digestion model setup to mimic the animal species that we're testing in, and that has also led to better in vivo and vitro correlations. The last that I put in here is that also a tendency towards designing specialized population digestion models, and here there has been some focus on the pediatric population, but that doesn't really solve this digestion absorption problem. It just means it's a way of doing in vitro studies that are more relevant for that population, especially in the case where ethically we cannot do so many in vivo studies. So therefore these models are getting more attention, but these two are really the ones addressing the issue with the lack of in vivo and vitro correlation. With that, focus on this and on to Mette.
Cool, thanks. Yeah, so with the specific study that we did in with using all of this knowledge and everything, we focused on trying to improve the estimation of absorption from all self-nanoemulsifying drug systems through combining intestinal in vitro lipolysis and permeation. So you might recognize some of the figures here. Here we have the in vitro vessel that we usually just use. However, this time we have taken samples and then put them into this permeative step. So we have the in vitro model and combining it with advanced diffusion cells equipped with this the PermeabilitySeries barrier. For this, we have in the PermeabilitySeries barrier, we are using the acceptor compartment as the lower compartment, in which we have PBS buffer with BSA inside it at pH 7.4, also having temperature regulation and stirring inside. Before we are running each of these systems and having the samples taken directly from the intestinal lipolysis vessels, we are then having the PermeabilitySeries barrier vetted with blank medium beforehand, with no permeation marker in it, no drug, no whatever. Just simply to have it vetted before, then later on adding on the lipolysis sample. And the samples will be taken over time, and inside the medium, we specifically for this project, we added calcine as a permeation marker inside the medium itself. And that way we are able to analyze the drug permeation from the formulation that is being digested during the lipolysis, and also from the permeation marker calcine simultaneously during this setup. And we had some different focuses that we specifically wanted to specify when we were trying to set up and develop this designing this model. Specifically, we wanted to use existing equipment, something that we already had in-house, but also something that other people might have lying around in the laboratories, so that they would be able to use it as well. And we also wanted to look and see if there was any time-dependent effect of lipolysis on the permeation of the drug that we tested. Thus taking samples before adding these digestive enzymes, and then during 15, 30, and 60 minutes of digestion, seeing then individually after this time how much digestion would have affected the permeation of the drug that we were looking at. Also, we were trying to have an absorption surface area to donor volume ratio closer to that of the human intestine. And this is because some studies have been pointing out that many in vitro models that are usually just focusing on permeation have an A/V ratio of less than 0.5 per centimeters in the in vitro systems, which is way lower than the 1.9 to 2.3, which is observed in the human intestine. And so in our current setup, we were able to add a larger volume that normally would be possible, thus having a A/V ratio of 1.34, which is then not the same, but at least closer to that of the in vivo situation. And lastly, the reason we were having BSA in bovine serum albumin, in case you don't know it, in our PBS buffer in the acceptor compartment, was in order to ensure sink conditions when we were doing these experiments. Since these are poorly water-soluble lipophilic drugs that mainly would be the focus of such a setup. And in order to further on evaluate the model, we had to kind of test it. So we chose a reference study from our own group, in which we had Senarison, which is a poorly water-soluble antihistaminic drug, and this was incorporated in some different SNEDDS formulations. So you can see the blank SNEDDS up in the corner. Here, the composition, but specifically Shiro Shikira and her colleagues and everything used 5 different formulations and tested in the system. We had SNEDDS formulation at SNEDDS 80%, so it's Cenericin dissolved in the SNEDDS formulation at 80% of solubility equilibrium in the SNEDDS system. We have the super SNEDDS suspension, in which Cenericin is added at 200% of this solubility equilibrium solubility in the SNEDDS, and also a super SNEDDS solution, which again is 200% of the solubility equilibrium, however, in solution. We have the chasing principle, which we have the lipid load of a blank SNEDDS being added just before an aqueous suspension. So this formulation would then in total be equal to the SNEDDS 80%, however, in kind of like in two parts, and then an aqueous suspension in the end. And these formulations were then tested in this one vessel lipolysis system, in which you can see the authors then saw that all of the SNEDDS formulations you can see up here in the top were kind of performing equally, and also all of them ready and in the aqueous phase, and would be assumed ready for absorption when giving in vivo, and all of them very much outperforming the aqueous suspension itself. But this might not be interesting if this didn't happen. That they obviously saw that when they're testing the formulations in vivo, they saw a completely different picture. So that in this case, for Cenericin formulations, these two formulations, the SNEDDS 80% and this chasing principle, were outperforming the other three formulations that were otherwise performed equally, which is completely a different picture to what was observed in vivo. So that's why we wanted to take the study and then use it and test it in our newly designed in vitro lipolysis permeation model, and testing this on three criteria: whether or not we were able to replicate the lipolysis systems data, in vitro data on that, whether or not the PermeabilitySeries would be able to maintain its integrity and stability during this setup, this combined setup, and whether or not combining lipolysis and permeation would be able to estimate the oral absorption of these 5 formulations that were tested. And for this specifically, I guess you are mostly interested in the last 2 points, so those are the ones that I'll show you just now. And here is also a picture you might like to see why calcine is actually also a nice practical marker to work with, because it's very yellow and orange. So this is after an experiment I was running for 3 hours, and you can see here that the lower compartment is turning more and more slowly. It has been turning yellow during these 3 hours that we've been running the permeation. So normally you would have this covered in foil, aluminium foil, but that wouldn't make a nice picture. But this is kind of like a showing of our stability of the apparent permeability of calcine that we ran during our experiments. Here we are having a control where there was no formulation added, giving us an internal control in the study of how the setup would affect the permeability barriers and the calcine permeability. And this value was found to be similar to the one that has been reported by Hannah Dabibi in 2017, when she tested a similar setup, also with SNEDDS and calcine as a marker. So when we're trying to compare our control to when we were running our different formulations in our combined setup, we saw no significant difference between the permeability of calcine, and we saw a slight tendency, but not significantly so, that there was a higher permeability of calcine when we are having these digestive enzymes included in the model as well, which you can see by having this without digestion enzymes and then increasing times of digestion enzymes. The only obvious exception for this is the super SNEDDS solution, that you can clearly see it has a way higher calcine permeability than everything else. So we were kind of worried about this, but however, it also showed that it was still lower than if there had been no lipid barrier in the PermeabilitySeries, which Hannah Dabibi also published in 2015. So this value I just put in here, which is then higher than the value that we found, making us assume that there's still some barrier integrity left in the PermeabilitySeries, even though we are exposing them to these conditions. And it was also confirmed when we were looking at the narration data that was recorded simultaneously. And here you can see what you would get if you were setting up, having the system set up. You get all of these permeation profiles, and like I said, we kind of want to see if there was any dependent effect on of the lipolysis on the permeation. And there's a lot happening in these graphs, but you might be quite able to see that there's not really any difference between for all of the formulations between the different time points that we've taken samples out from the lipolysis vessel itself. So we concluded that for this drug and for this formulation, there was no time-dependent effect of lipolysis on the permeation, and then we pooled them together when we were trying to do the further in vitro in vivo correlation comparison. So therefore, we have now here is a little bit more clearer picture to see. We have the formulations where they then pulled together within itself for the different times, but having the SNEDDS 80%. And chasing principle outperforming the other three formulations that were otherwise equal, which was also what was observed in the in vivo data that I also showed you just before. However, there's the difference that is quite obviously to see is that the chasing principle didn't perform similarly to the SNEDDS formulation, which is contrary to what was observed when we had the in vivo data. However, the ranking is still, if you group them together with the lipid load, then you still have the two formulations with the higher lipid load, so the SNEDDS and the chasing principle, outperforming the other three formulations. And when we are then having the AUCs in vitro against the in vivo AUCs plotted, then we also get a linear correlations of 0.92, which is then making us more confident in our model itself. As you can see here. So overall, we were able to estimate the order absorption of the 5 formulations by rank order and linear correlation. However, there's no doubt that further studies are needed with other lipid-based formulations to truly evaluate the model and also figure out whether or not the lipolysis itself will have any influence on other drugs and other formulations. And then I guess this is the end of the talk, and we have further reading possibilities in the review we wrote 2 years ago, and also in the study, the paper we just published a couple of months ago with this study specifically. So if there's any questions.
Perfect. I think you're through with the presentation, and we already got two questions. The first being, could you have used another type of barrier for the model, for example, PVPA, PAMPA, or KCO2 cells?
Yes, we can answer that. In principle, yes, you can use the different types of barriers. And here it's important to know about the variations in the barriers. And this has also been presented by Max in a previous, that there's different characteristics of the different barriers. And if you have a drug that you know has an active transporter, for instance, using a KCO2 cell model will be a better simulation of the intestinal absorption. Using KCO2 cells is a little problematic with digestive enzymes though. But I know that at least in Uppsala, they've done some studies where you can sort of change the setup a bit in a way so you can have some digestion occurring and protect your KCO2 cells at the same time. So it is possible to use that. And we've also worked with PAMPA membranes that actually also work fine in this setup. The general thing you have to keep in mind is that, however, is that when you're doing digestion studies, we are digesting lipids. So if you have lipids in your barrier systems, you might also digest your barrier. So therefore, it's very important to include some sort of marker of your barrier integrity to make sure that you still have a barrier after your digestion study is done.
Okay, perfect. The next question also is about the digestion, and it is asked if digestion is necessary at all for the digestion step.
Well, Yes, and based on this system and what Mette just presented, we can see that here in this specific case, we actually saw no impact of the digestion. So doing the permeation step directly after just dispersing the formulation in an intestinal media gave us the same results in terms of rank ordering the formulation than having a digestion for 60 minutes, for instance. So in this case, you could discuss that maybe it's not relevant. However, it will really depend again on the drug and the type of drug delivery systems. And already now there's studies showing that you do need this digestion in order to get a good correlation in vivo and vitro when it's a different formulation. And there it's in Tromsø, I believe, that there are a publication out where you can see the necessity of mimicking the digestion. But like I said initially, you always have to sort of consider what's the specifics of the drug and the digestion systems. How many things do you need to mimic in vitro in order to get a good correlation to in vivo? So that really depends on the specific system.
Mm-hmm. And what volume do you withdraw from the digestion step up to pour into the permeation cells? Do you replace the volume by a fresh medium?
No, I don't. So we, well, I have to recall. We added 1.5 milliliter to the barriers themselves in the fan diffusion cells, and then we had we withdrew 3 milliliters and didn't replace any of it into the lipolysis vessel because it's a very complex system. So you're both taking enzyme, formulation, and media up, so it would be very difficult and impossible to replace. So we generally in lipolysis methods you don't really replace during this, and I think it was either 2.5 or 3 milliliters that was the sample volume itself, because some of it was used to quantify the different phase separation that was occurring in the lipolysis vessel in order to compare with the reference study that was already done.
Mm-hmm. And someone asked if you can tell in advance with which kind of compounds a simple lipolysis model is not working.
This is a very good question, and I wish I could tell you the answer. So far, it's a no, and of course that makes it more problematic. But based on like the recent review, and also we did a review 2 years ago, and Fini did also a large review 3 years ago or something like that, and there's been a lot of like trying to collect all the in vivo and vitro data and and looking for tendencies. So, for instance, Powson has this lipid classification systems, so that if you have a lot of lipids, you have a classification 1, and if you have a fewer lipids, then it's a type 3. If there's more surfactants, it's a type 3 or type 4. And there's like, maybe we can say that if it's a type 1, lipolysis is more important, and if it's a type 3, then you need the permeation. But so far, actually, we haven't found any trends, and neither have the other people writing the reviews. So maybe I'm really hoping that at some point we can figure out what is the decisive point. But right now we don't know.
Okay, something for the future to come, hopefully. Someone asked if it is possible to carry the permeability assay using PermeaPad and not detecting the active compound in the receptor cells. What could be the reason then?
If you can't quantify the drug in the acceptor side?
No, in the receptor side.
Yes. Yeah, but I think then that is the most common reason is that you only permeate a small fraction of whatever is in the donor side. And as Mette also said, that especially in these cases where it's poorly water-soluble compounds, the solubility is going to be really low. So then we have very little present. So often it's because it's too diluted. And then one thing is that I know also has been presented in this series before is concerned with like minimizing the receiver volume so that you can sort of up concentrate, or adding some sort of surfactants that can create a better sink so you can have higher concentrations there. And the later part was what we did in this study in order to have enough drug permeating and enough drug to quantify.
Another question is if there is maybe an advantage to use an oil-soluble fluorescence marker instead of a water-soluble one to follow the experiment.
I don't know. Maybe. The main reason we chose the fluorescent marker was so that we could quantify by a different assay, so that we could have something with UV detection and then at the same time something with fluorescent detection. But whether it's hydrophilic or lipophilic, but I think like in general, if it's usually we use a hydrophilic marker because that doesn't want to permeate if the barrier is intact. So very little is going to permeate. But if you have a very lipophilic marker, I'm assuming more of the marker permeates or goes into the membrane, and therefore at least it's not really a marker for an intact barrier. So I think it's more common to use a hydrophilic marker, at least for the integrity of the barrier. But if you have something permeating a lot, you can also use that as a control, sort of to see this is the level it normally permeates at, and if that changes, then something has happened to the barrier.
Another question coming from the permeation side is if you see any limitation by the particle size.
I think that in our group, we usually work with small molecules, I have to say. So we haven't really seen much problems with the different permeation barriers and particles. I've done studies with Cago2 cells because they're so sensitive that if you have a lot of drug particles present, they have a tendency to disrupt the cell layer because all the drug particles are gonna like lie on the cells and then they die. So in that case, the number of particles at least matters.
Yeah, I think there is a specific number of particle size that can permeate through the permeapad barrier, but I'm not quite sure, but we can hand over this information later. Another question is how long this particle was stable in the media. Don't they were destroyed or sedimented?
Like the SNEDDS emulsion droplets, like in our digestion setup, we have stirring all of the time, and that's of course like we usually have an overhead propeller stirrer, and that's not super biologically relevant because we don't really have a stirrer like that in the stomach, but that or the small intestine, but that is really to ensure we have a homogeneous solution for sampling. So as long as we have stirring, we won't have any sedimentation, and these particles are going to be separated if they already have formed this nice emulsion. But in this specific combined lipolysis permeation model, one of the drawbacks that we have already listed is that we don't have any stirring in our donor compartment. So when we add this digestion sample to the Fran cells, we only have stirring on the acceptor or the receptor side. And that might, of course, mean that maybe there's sedimentation. Something might happen at the donor side. And right now, we don't know what actually happens and to what degree sedimentation might occur. But what we can see is, like Mette described, is that we don't see the barrier's integrity going down, and we see this very linear drug permeation profile. So looking very much like a steady state flux. And I'm assuming that if the donor media changes a lot in composition, if all the particles would aggregate or something like that, we would see something occurring in our permeation profiles. So sort of indirectly, I'm assuming that it's not a huge problem because our profiles look like it's a very steady state permeation occurring.
And yeah, we have two more questions. if you can use this dual model for mechanistic understanding, transcellular or paracellular permeation.
If we can use the model for seeing if it's transcellular or paracellular?
Mm-hmm.
Then I would do with the CACO, right? Or some different cell-based.
I think if it's for the mechanistic understanding of the specific permeation, I feel like it gets very complicated when you also have the digestion process in. So at least I would do more simpler permeation studies initially to sort of get a mechanistic understanding of how the drug permeates and which parameters affect its permeation before adding on this digestion and lipid-based formulation also. But I think in order to distinguish the type of permeation, you need models that can mimic that part, at least. And I don't think, as far as I know, with the PermeaPad, that even though it's mainly a transcellular diffusion, it's mimicking or diffusion. You might also say it partly captures some of the transcellular, but it can't really distinguish between how much is transcellular or paracellular. And for that, usually you would do some sort of a CACO2 cell setup where you can have add a drug that might open your tight junctions. So you increase this paracellular transport and then look at the impact that has on your general transport. And then you can sort of play around with seeing if you change the ratio of paracellular to transcellular, then you can learn something mechanistic about how the drug is absorbed.
Coming back to the pore size, someone says he assumes that the PermeaPad has pores of a specific size. Yes, that's correct. And he asked if you need to make sure that the resulting micelles in the donor chamber has a bigger size than the size of the pores.
I'm assuming that they will have a bigger size. At least I feel like didn't we do studies without the lipids in with just a filter? No. We tried that not with the PermeaPad then, but then with Pamba membranes, we tried doing permeation without any lipids in it to see, because then it's just the pore size, and then it permeates completely different than if you have the lipid barrier present. So I think the lipid barrier really is what matters most for the permeation of these systems compared to the pore size alone.
Mm-hmm. And now the last question is, maybe she missed the part, but what do you have to draw samples out of the lipolysis model and put them in the permeation model? Isn't it possible to combine both of them?
If for the study we did here, we took samples from the digestion vessel and transferred them to this French cell and then did the permeation setup. In principle, it is possible to do everything at the same time. But the main thing is that when we have, if you want digestion, controlled digestion, so that by measuring pH and controlling the pH and the degree of digestion by having titration, you need a rather big volume on top of these permeation. Maybe I can find it. Picture of the model. Oh, I'm so slow. I don't know.
Almost there.
But the part is that if you want everything controlled, the digestion process, you need a large donor volume. And if you want this part to take place here, you need a big volume here. Otherwise, all of these things cannot fit. What?
You need to share your screen if you are showing something.
I thought I was already sharing my screen. Then I will just try to share it. I'm sorry.
No problem.
I'm really bad at this.
You got it already. Oh.
I can't see what I'm sharing. Am I sharing my presentation now?
Yes.
Yes was the answer.
I hope I am. But
Yes.
apart, if you want this digestion part to occur in the donor vessel, and if you want to control, so we know how much digestion occurring, and we need the pH is stable, and we have a homogeneous mixture with stirring, we need a rather big volume here. And if we increase this volume, then the volume to surface area ratio is going to go down a lot. Because otherwise it's just, with the setup we have right now, we cannot have a big enough surface area for this volume to keep this high surface to volume area. And in vivo, we really see that because we have this huge surface area in the small intestine, we have a really high ratio, so it's very little fluid spread over the whole intestine. And based on whatever publications has been out so far, it seems that mimicking this ratio between the volume available and the surface area is really crucial to get a good understanding. And therefore, it's sort of like, if you want to control this on top here, you lose the good or the high surface area to volume ratio. So it's sort of a trade-off what you want to do. But what you can say is that also in this setup, when we are transferring a sample here, then we didn't inhibit the digestion enzymes or anything. So we still have digestion occurring here. It's just not controlled anymore in the manner that at least we don't know how much digestion is occurring, and we don't know what the pH is anymore. And what's also been stressed already, we don't have stirring, so maybe all the particles sediment. So it's sort of a trade-off, but what you want the most.
So with an eye on the clock, we almost reached the hour mark, and I think we're quite through with almost all questions. And like I said before, you can also ask questions and send us an email, or Mette and Rana also an email. Thank you both for holding this presentation. It was very informative. Thanks for answering all the questions, and yeah, thanks for everyone attending the presentation. We have now uploaded the handout, so basically the whole presentation, and it would be nice also if you take part as a survey and help us improve the next presentation as well. Again, thanks both of you. Thanks everyone, and have a nice day, and see you next time to the next webinar. Bye.
Thanks.
Den røde knap, der kommer op en gang.